Hello everyone, welcome to another experimental biochemistry class. Today we are going to study about protein folding and denaturation studies. So basically in the previous experimental part we have seen that protein gives an UV absorbance and also fluorescence. Basically this is due to the protein residues, especially the aromatic residues present in the protein. Now what happens, basically protein stays at physiological condition within our body, so it retains its structure, and when it is subject to some sort of stress like excess acid or some denaturating agent or temperature, something like that, it denaturates. What denaturation means? Basically it means it loses its tertiary structure, that is the entire 3D structure of the protein breaks to some extent, partially or fully. It determines the degree of denaturation. The details of denaturation you have studied in the theoretical classes. Now we are going to see how to basically experimentally observe protein denaturation. Now various ways are there in order to monitor the denaturation process in protein. Now the process we are going to adopt in this case is spectrometric studies. Mainly we are first going to observe the absorbance study using UV spectrophotometer. There are various types of denaturating agents which are commonly used. Mainly they are, one is temperature, the other is denaturating agents like guanidine hydrochloride, urea. So we will start initially with urea as a denaturating agent. As we very well know that urea basically denatures structure of the protein, breaks its structure, and thereby the change in absorbance of the protein can be noted down. So let us see whether the structure of the protein has any role to play on its absorbance value. For that basically we need a urea solution, and the protein we are going to use is human serum albumin, the protein having one tryptophan residue, and we are going to monitor the change in absorbance of the tryptophan residue. We have taken an urea solution. So urea solution we have prepared around 8 molar urea solution. However, the concentration you may vary. Generally for denaturation the maximum concentration we are using generally in biochemistry labs, it is around 8 molar urea. However, in this case since the maximum concentration at 8 molar, so we can't reach this concentration, we will keep our concentration up to 5 or 6 whatever we can manage. So we will initially take this urea solution prepared in a volumetric flask in a small beaker. This beaker should be initially washed cleanly and dried. Small amount of urea that will be required for our experimental purpose. Before starting our experiment, we have to initialize the UV spectrophotometer and do the baseline correction. Now initialization has already been done in this instrument. For baseline correction, as we have previously seen, that we need to fill these 2 cuvettes, that is the UV cuvette with water, which is the solvent in our present case. Now as you have already seen in the previous video, that the cuvette has 2 sides, one is the clean side where a transparent side, and the other is actually the frost side. We have always to touch this frost side while taking the solution. Now we will take water, that is in the pipette, and fill it properly before using, and preferably it should be dried. In this case we require 3 ml of double distilled water. Similarly I will be filling another cuvette with double distilled water. Now once you have filled this cuvette, you have to wash this transparent side properly, and I will put this in the chamber. Now this is the UV chamber. Here is the compartment, and this the holder for the cuvette. This has been previously discussed in the last video. Now when you are placing this cuvette, you should keep in mind that this transparent side should face the passage of light. This light travels from here to here, so the transparent side should face here. This one is for reference, and this part is for sample. So we will put this cuvette in the reference part, and we will take this cuvette. This is the sample cuvette. Currently it is filled with the solvent, no sample is present here, and this will be used for baseline correction. So we will keep this in the sample compartment, and then we will close the lid. So we have kept the 2 cuvettes within this UV chamber. Now this is the softer, as we have seen previously. First we have to do is to go to this method part noted by M. This is the method part. Click here, and here comes the spectrum method, and in this part we have to change the wavelength, and here it will start from 200 and this has been changed to 600, and it was initially a different part, but for the current analysis we will require 600. The scan speed is medium, sampling interval 1 nanometer wavelength, reading will be taken. The file name we have currently set up in under NPTEL denaturation, and other parameters are more or less same. The slit width is 2, and here let us click OK. Now, if you want to change the wavelength range, and you might change this part, but currently since protein we have to analyze the absorbance around 200 to 300 nanometers. So, it is better to stick to 600 nanometer. So, let us click OK, and now we have to go to this baseline. Initially we have to do baseline correction. This is the baseline part just next to auto 0. Let us click this baseline, and it will prompt the another option where you can change the start and the end wavelength. This has been set 600 and 200. So, let us click OK, and the machine will slowly initialize itself for baseline correction. In this part you can see that the wavelengths are being scanned here, that is the baseline correction is being scanned from here to here. Do not do anything, do not press or go to any options while baseline correction is going on. Now, it has slowly reached to this 200 nanometer, and the instrument will be ready soon. Here we can see again these options are enabled. So, our baseline correction is done. We can start with the experiment. Now, we have to see the change in the absorbance spectra of protein in presence of urea. However, before that let us check how the absorbance spectra of urea looks like. Okay. For that, what we will do, we will take urea solution in the cuvette. Now, in the cuvette we have initially a water. So, we will take small amount of water out from this cuvette and fill this with urea solution and mix it so that the solution becomes homogeneous. Please see that air bubbles are not formed within this cuvette. Now, once we are prepared with this urea solution in the cuvette, we will see the absorbance of this urea solution. So, we have taken the urea in the sample compartment. Now, we will do auto 0 initially, so that the value at 600 nanometer becomes 0, absorbance value. 0 is done, we can see the absorbance is set to 0, and now we can start with our measurement, and let us click start, and here we can change it. Let us change this value a bit, around 0.5. So, here we can see here is a peak at around 205 nanometer. From here we can see no remarkable peaks are obtained in this region 200 to 300. So, let us check this one by lowering this value around 0.1. We can see this lower range. Here we can see some arbitrary spectra are like there, but it is not a smooth in peak are obtained out here. So, we can say that there are no significant peaks in the region 200 to 300 for urea, and this peak might overlap with the protein amide bond peak. However, now let us check if we add this urea in the reference compartment, what happens basically. For that, we will add the same amount of urea in the reference compartment. So, let us first save this data as urea 1 in a spectrum file, and you may also save it as data text file, denaturation part. Now, we have added urea in the reference compartment, and let us see what happens. We will explain why you have added it in the reference compartment. Now we have to click this auto zero. Now let us click start. So the scan has already started. Out here we can see that the wavelength scan is being taking place. We can change for our convenience if we want to see the spectra. It gives an absorbance value of around 207, similar to the last one. However, here one important thing to note down out here is that we have seen that this black line indicates only urea, and here the blue line basically indicates urea when added in the sample annual reference compartment. What this basically does is that it basically nullifies the effect of urea in both part. The small absorbance out here basically may be due to the manual error which might have occurred in while taking the solution. The volume may not be exactly same, or the concentration might differ a bit. So this small range of error might occur in the lower wavelength range. However, since we are not much concerned in this case, so what we will do, we will just consider out here in the 200-300 region where we can see no significant peaks have been shown for urea. So we can easily carry out our protein denaturation studies. Now we are going to start with our actual experiment, that is the change in absorbance of protein. The protein we have taken here is human serum albumin. The human serum albumin is basically protein known as HSA. Now HSA has one tryptophan residue, and as we have discussed, we will monitor the change in the tryptophan absorbance. Now this HSA, we are going to take a small amount of HSA in the cuvette, and we are gradually going to add their urea and see how the absorbance spectrum changes. Now this is the cuvette. Out here we are taking small amount of water in the cuvette and filling equal amount of HSA solution out here. Now please remember to wash this cuvette properly. After this previous observation, I have cleaned this cuvette very well. This cuvette should be properly cleaned and if possible to be dried, and then we have to take the next solution. So we have taken HSA, we are going to homogeneize it as much as possible. Try to avoid bubble formation. Bubbles are being formed out here currently. Now we have to just remove those. You can remove the bubble with cuvette. Try to mix them homogeneously, and the bubbles are being removed. Okay. Now this is HSA solution. We have to see the absorbance of this HSA solution. So we have taken HSA in the sample cuvette, and the reference cuvette is currently filled with water, double distilled water. Now we can see some sort of absorbance is coming out here. So we have to do auto zero. It will take a few seconds. Now here we can see the absorbance is currently set to 0. Now we can easily start this experiment. You can see the scan has already started, and whenever scan is going on, it is recommended, or rather it is advised, please do not open this lid. Now, here you can see a gradual appearance of the spectral line. Here we can see the major peak of the amino acids, and there goes the spectra for the peptide bands. Now, this one here is the region of concern. Now, the first thing we need to do is to save this part, denaturation. You have to save this one as HSA 0 urea. That is, we haven't added urea, this is the normal HSA, so 0 urea. So, we are saving this as spectrum files, and after that we are going to save as in the data print table.txt. We are saving it as a text file with the same name, 0 urea. Now, let us see how it comes. Basically, if we drag this line, vertical line, we can see around 278 we are having a value 0.091, and however, if we want to see, or if we want to study what is the value of around 280, which we can monitor, we have to just click here 280, we have to just enter 280 out here and press enter, and it will give the value of around 280. So, you may consider anywhere around 278 or 280 depending upon the situation, which I will discuss later on. So, this is the HSA solution we have just taken out from the sample compartment. Now, we will be adding urea to it. The urea will be around 0.2 molar urea, the concentration currently set out here. So, this is the urea solution. We are going to take definite volume, so that the concentration is 0.2 molar, and we are going to add it out here and mix it properly. Try to make it homogeneous. See, no bubbles are there. Wash the two transparent sides with the tissue paper, and let us transfer it in the chamber. So, let us click auto 0, although you can see the absorbance out here is all set to 0, but we can again do it auto 0. Auto 0 is complete, and now we will start with the scan. Let us click start, and the sample scan has already been started. Here we can see the absorbance slightly increases from the previous one. Now, once complete, let us save this one, and then we will go for analysis. To save in denaturation, now you can click here HSA underscore 0.2 molar urea, click save, and then go to save. In the same name, save it as text format. Okay. So, now coming to this analysis, what is basically happening out here, we can see if we change the scale a bit around 0.5, here we can see the green line is basically for protein without any denaturant or urea, and this red line is when 0.2 molar urea has been added, and we can see there is a change in the absorbance value out here. So, what we need to know is that we basically need to analyze this change, where the change is actually occurring. Now, the change we can see is occurring mainly around 278, the maximum is observed around 278 out here. And then what is the intensity of this red line, we can consider from here 0.214. It is recommended to currently write down this intensity whatever you get for a particular concentration. However, if you miss intensity of the first one, say if you did not note down this for pure HSA, what you can do is that you can click here, these are the options, you can click out here, this is the raw data HSA 0 urea, double click out here. Once you double click, this will be selected, and you can go to this active part. Out here we can see active overlay stacked, and if we click the active part, Now the active part will be shown, the part will be shown which is activated, that is only HSA. Here we can see at 278 the value is around 191 point, sorry in the activated part. Here we can see the HSA value, the absorbance value is 0.191. Now if we click this HSA 0.2 molar, here we can see around 278 the value is around 0.214, that is from 191 it is increased to 0.214. So let us click on this overlay, and what does stacked do? The stacked actually shows you different types of spectra one above the another, it is stacking them, and it is better to analyze in the overlay part where you can basically see the changes in the absorbance. Now we will add another concentration of, now we are adding the next urea to the solution. For this we are setting the concentration 0.4. So we are adding the requisite volume of urea to this HFA solution. Now there are two ways of adding. One, you can just take this pipette and directly add it out here, kept in the sample holder. Now we can add it out here, but however it is recommended not to do that, although sometimes people add out here, but it is recommended to take this cuvette out. So we have taken out this cuvette from the compartment and we are adding the required volume of urea to it. Please note that change the tip of this pipette from the last urea solution you have added. Never reuse a tip, because the solution of urea you have added here, you have dipped this tip into the solution. So what happens, it has been pre-contaminated with protein as well. So never immerse this tip once again in the urea solution. So we are now making this homogeneous. Now why it's better to add this solution not in the chamber and but rather outside. So what happens is that when you take the solution or any solution and add it in the chamber, sometimes it may happen that the solution might not fall in the cuvette, or it might get spilled and fall outside. So it is a very costly instrument, the spectrophotometer. So it is better to avoid any sort of liquid contamination with it. So better you take out this cuvette and you can basically control it while you are adding. And now we have made it quite homogeneous. We will remove the sort of bubbles formed out here. Now we will go for the analysis. So now we will do auto zero. Each time you take a reading, perform this auto zero part. Okay, so it is currently again set to 0. So its value is coming 1. So once we are again doing auto zero, somewhere this value, yeah, it's now 0. So let's start. Now let's save this one, save as. We can click here and the name will appear, and we can change this name to 0.4 urea and the spectrum file. Another file will be saved in the name of 0.4 urea, and again we can click the save as, go there, we can change to data.txt data file and again save. Now coming to this part, what we can see, if we change the scale to 0.3, here we can see the shift, or rather the increment is quite low out here, because here the line above the red line, the gap is not that much. We can also visualize the gap by lowering this or increasing the lower value. Upon increasing the lower value to around 0.15, here we can very well observe the change out here. Now this part, this is the green, this is the red, and the part just above this red line, this is a black line, this is for 0.4 molar urea. Basically what's happening is that upon giving 0.2 molar urea, the denaturation has started, so the absorbance increases. But 0.4 molar of urea, the denaturation process does not vary that much from 0.2 molar of urea. So the change in absorbance is not that much prominent. Now let's add another concentration of urea. So, what basically has happened out here, that this part has gradually decreased from the previous part. This may be basically due to the dilution process. Since we are adding it to HSA, we are basically doing a dilution, that is we are adding urea constantly to it. So, the concentration of HSA might be diluted, and that is why we can see a gradual decrease in the absorbance value, or fluctuations might occur out here. So, it is recommended to subtract the control after you have done the experiment. I will show it once. Now, we have to save this one, save as, one molar urea, so let us zoom it a bit, 4 0, or it is around better to say 0.15. So, here we can see there is a green line, above this green line this again decreases. So, basically the decrease should not be there, it is basically the dilution factor, the solution is getting diluted. So, that is why the absorbance decreases. However, once we subtract the control which is having equal amount of urea in it, then we will get the actual solution. Now, we have taken 1 molar urea, doing the auto zero. Now, it is done, let us start. Here we can see there is an increase in the peak intensity. This is for 1 molar urea solution. And we can calculate the intensity at around 278. Now, as we have discussed earlier, basically you can take the intensity at 278 or 280 wherever you feel, but you have to note down the change where the maximum change occurs. Generally, the maximum change might occur around 278, or the maximum change might occur around 280 or 281. Basically, on 270 to 280 where the maximum change between this HSO without urea and HSO with urea occurs, and there you have to take the change in the absorbance value. Now, we will go to 2 millimolar. Now, we will go to 2 molar urea solution. We have taken for 2 molar urea solution. Here we can see the change again for 1 to 2 is not that much as for 0.4 to 1. So, we can see the change once again coming to 0.3 out here, and this red line, this part is basically for 2 molar urea. Now, we will be adding 4 molar of urea. So, we have done auto zero, again this is for 4 molar. And start. At least you should take 5 to 6 reading for this plot. This plot is the concentration of denaturant versus the change in absorbance at 278 or 280 wherever it comes maximum. Now, here we can see it is for 4 molar urea. Now, we are doing it for 6 molar urea solution. The effect of urea of a concentration 6 molar on HSA protein. Here we can see the intensity increases, the absorbance of this HSA solution. So, we have seen how the change in concentration of urea causes the change in absorbance value. With increase in concentration of urea, the absorbance increases. Basically, what happens is that upon adding urea, the protein unfolds and the tryptophan residue comes out in the medium. So, the extent of tryptophan residue exposed in the solvent medium increases its absorbance value. Actually, it was folded, that all the tryptophans were not exposed, and tryptophan residue is basically buried in HSA, and once it opens up upon adding a denaturant, more and more tryptophan residues of different proteins in the solution are being exposed. And now as we increase the concentration, two things occur: more and more proteins are getting denatured, and also the extent of denaturation increases, and the extent basically increases maximum at around 8 molar of urea, which we are not currently performing here. We are restricting ourselves to around 6 molar urea. The reason is that we have prepared the solution of 8 molar, and now we cannot actually achieve the solution 8 molar in the cuvette, since the stock solution is 8 molar. Anyways, so what is happening is that as we are increasing from 1 to 2 to 4 to 6, we can getting an increase in the absorbance value, and the increase in absorbance value is much higher. So what is basically happening is that at higher concentration, the protein is unfolding at a maximum rate, and at this way what is happening is that more and more tryptophan residues are getting exposed, thereby increasing the absorbance value. Now we are going to see the temperature effect on protein denaturation.